I think we have seen enough of meiosis, so let's see some Mendelian inheritance and how it connects to the meiosis. But before that, I need to describe something called gene pool. Gene pool is a hypothetical concept that we use in genetics a lot. Uh, it represents a population, and all the alleles in a population is pooled here, and in every time an embryo is formed, we are actually randomly selecting from this pool. Okay, first of all, what is an allele? An allele is a variant of a gene. And how do they arise? Of course, by mutation. Because the same gene can mutate, it can create some hyperactive version, an inactive version, a less active version, a version that interacts with something else. So, and there's no upper limit for the alleles. Some genes can have just one allele, as in here. They can have two, as in here, or they can have eight, as in here. It depends on how many mutations they accumulate in history or in that population. So each time an embryo is formed, we are going to randomly select out of this gene pool. So in this embryo, we are going to select two alleles for the gene A. Let's say it is A1 and A4, and two alleles for gene B. Let's say it is B2 and B2. I mean, this is heterozygous. It's heterozygous for this point, homozygous for this point. It doesn't have to be heterozygous. For the C, let's say it is C2 and C3. For D, let's say it is D2 and D2. It's very likely to be homozygous. For the E1, it has to be homozygous. Anyway, there is just one single allele, F1 and F2. And it's very difficult to be homozygous for the G allele, G gene. Let's say it's G5 and G8. Okay, so this is just one of the possibilities that will be generated in this allele, in this population. Each time an organism is fertilized, each time an animal is going to grow, it just selects this too. So the larger gene pool means higher variation of progeny. And we know that diversity, variation is the power in biology. So a population is only as strong as its diversity. Larger gene pools are usually better because whenever there is an unexpected pathogen, I'm not going to say coronavirus because it is not that little, but this unexpected pathogen can be Ebola, which is like 90% lethal. Some of them will just survive and beat with the new pathogen, or they're going to adapt these new conditions with the help of these new combinations, because one of the combinations will be successful enough to beat in the new condition. Okay. But to do these calculations, they are all genetic calculations, and genetics is highly related to mathematics and statistics. We have to understand the basis of Mendelian genetics. So let's go back to Mendel and find what Mendel found. Before Mendel, of course, everybody knew that there is an inheritance, which means we look like our parents. What people didn't understand was that they thought we inherit our features only from our father. Well, that was a patriarchal, at least after the Sumerian culture, it was nearly always patriarchic. So it was a male dominant culture, and people tried to underestimate women so that philosophically they are going to have more power, or religiously they are going to have more power. This is one of the drawings in the sperm. A small man lies. So every feature, every information comes from the father only, and the mother is just responsible of growing the baby and has no contribution. So what Mendel proved was the opposite. He proved that when there is a new baby, both parents equally contribute. So this was kind of revolutionary. People didn't want to believe it. or at least didn't want to believe that it works in humans, because maybe it works with the dogs because people had experience with mating the dogs, but they didn't want to believe that the same rules applies with the dogs and humans, because dogs are kind of stupid animals and humans were clever. They have high values, but it turns out that they were under the control of the very same rules, which are called the Mendelian laws. And it's not only humans and dogs, actually every kind of diploid organism, including the plants, it applies to the same rules. And anyway, Mendel did not work on the animals, he worked on the plants, the pea plant. He chose the pea plant because he could see clear-cut differences in the phenotypes. He could see white flowers, purple flowers, okay, not something pinkish or light purple. And this was very useful because if you were working with humans, imagine that you can work with humans, you are living like 1000 years so that you can experiment on humans. Even then it would be difficult because we don't see many clear-cut differences between humans. The height, for example, is quite continuous. But in this case, it's binary. It is either green or yellow. It is either wrinkled or round. It is either inflated or pinched, or either tall or short. So we can simply classify the pea plant as tall and short. This is something that we cannot do on humans. This is something that we cannot do in other animals as well. If you were to do this experiment on mouse, even then it would be very difficult because you cannot divide the mouse as tall mice and short mice into two. Okay. This way, Mendel was kind of like you, or let's say he was really clever to choose an example, choose a model that is going to fit in his experimentation. Okay, what he did was that he crossed the organisms. True breeding means homozygous. Okay. He got the homozygous yellow pea and green pea, and then he cross-fertilized them, which means, remember the very beginning of our class, so this is a sexually reproducing organism, a flowering plant. There are male and female organs. So in one cases he cut the male organs, only female organ left. In other cases he transferred this male organ into the female organ. So he controlled their sexual reproduction, which again is difficult to do in humans because people wouldn't like the idea that you choose their mate, who they are going to have sex with. So this is homozygous, another homozygous, and then in a controlled way he outcrossed them, which means he didn't allow the hermaphrodite style reproduction, which is here. He got the eggs and sperms from two different organisms so that at the end they're going to have a hybrid genome. Of course, he didn't call it genome. He had no idea about the DNA molecule. He is not a geneticist. He is not a molecular biologist. He is not a biochemist. He was also educated by the physicists, so he was good at physics and maths rather. So he never had a microscope, a real good microscope. He had never, purified DNA molecules and checked how they move under the microscope. All he could see is the phenotype directly. So what do they look like? And he called them traits, the green trait and the yellow trait. And he was following these traits and the crosses. So all he had in his hands that how he crosses the organisms and the numbers in the progeny. Okay, the first thing he observed was that when he has the hybrid, which is called the monohybrid, because we are talking about one single trait, which is the color, the green feature is gone. The green trait is gone. Where the hell did it go? Did it just disappear? Because it quite looks like this thing, only the father determines the features of the progeny, the other sex has no contribution. It was surprising, but he didn't stop here at F1. He let the F1 to self-fertilize, so it was a hermaphrodite-like reproduction. And he realized that in the next generation, the green trait came back. So, first, this hypothesis is wrong, uniparental contribution. So, both parents, both sexes contributes. This is the first thing he has seen. Second, this green thing disappeared previously and then it came back, so it was hiding. The green feature was hiding here. Where did it hide? How come it hides? And another thing he realized, whenever he does this experiment, he has more yellow and less green. And when he did the maths, he realized it is always 75% yellow and 25% green. So it's a 3-to-1 ratio. So depending on his observation, he said that in every organism, I should take a little bit here, in every organism, there are two determinants. And these two determinants divide into two in reproduction. Okay, this is the old language, the end of 19th century. Currently, we know what these determinants are. Now we call them genes. Mendel didn't call them genes, he didn't know them. And he said that they divide into two. Now we know it is meiosis. Okay, so he actually observed meiosis in maths. And he said that when they divide into two, only one is inherited to the next generation, to the progeny. And we know that only one of the gametes will be used, only one of the alleles will be used. So this is the first Mendelian law. So I realized when you were writing your lab reports that you didn't really know the Mendelian laws. It's a shame. If you're going to be a geneticist, you should know the Mendelian laws as your name. So this is called law of segregation. Which means the alleles the organism has, they are going to divide into two. Okay? And because they are dividing into two, you can calculate them with a Punnett square. By the way, this is not called Mendel square as you have written in your lab reports. This is called the Punnett square because Punnett has made the square. So in the eggs, the possible gametes the possible alleles will divide into two. In the sperm also. This dividing into two is meiosis. This coming back together is fertilization. Again, the same old cycle, meiosis and fertilization, meiosis and fertilization. So how it happens, really? Let's write the genotypes. There were the yellow seeds and the green seeds. And because they are the diploid organisms, they have two copies. If this was a triploid or tetraploid organism, you could expect 3 or 4 different alleles, or maybe the same allele 4 times. But all in all, there would be 4 homologs. But pea plant is a diploid organism like human, so there must be 2. In the green, there are this small y. So in the meiosis, they divide into 2. So it's going to produce gametes. Well, actually, each cell is going to produce 4 gametes. But they will be all capital Y here, and they will be all small y here. So these are the adult cells. These are the gametes. Now, randomly, we are going to pick one of the gametes. Remember the analogy of gene pool? We randomly select one of the gametes here. And it is really just a matter of chance. But because the organism here is homozygous, there is no difference in the gamete. So whatever gamete you choose, you're going to choose a capital Y here and a small y here. Next, it is going to be fertilization. So these two are going to meet. So in the next progeny, in the F1, you're going to have capital Y, small y, which is going to be a heterozygous. This heterozygous is also called hybrid. Hybrid means combinations of something, like the hybrid car. It's a combination of gas consuming and electricity consuming engines. Now, this heterozygous will also go under meiosis, and again, it's going to create 4 gametes. But this time, there are 2 possibilities. 1/2 possibility is that you're going to get a capital Y gamete, and 1/2 possibility, 50% possibility, is that you will get a small y gamete. Now, this is where the things changes. This is where you get the gametes randomly from the gene pool. This is where everything has chances. Okay, and this is the point that provides the genetic diversity, because here you have different chances apply. Of course, these are the alleles that this organism has, and there are different organisms which are carrying different alleles. Let's see here, if we are talking about the gene G, there are 8 different alleles. So if you're selecting a couple of gametes from these 2, there are a lot of different combinations that you can have. So here there are only 2 alleles in the case of pea plants, so there are only 2 combinations. So let's make the Punnett square. So capital or small y from one parent and capital or small y from the other parent. Because this is a self-fertilization, the 2 parents are the same. At the end, you're going to have 3 different combinations. So one of them will be homozygous, 2 of them will be heterozygous, and this one will also be homozygous. So this is homo, and these 2 are hetero and identical. Now, here we come at the 3rd law of Mendel. Mendel's first law was the law of segregation. Mendel's second law is Mendel's 3rd law is the law of dominance. And it says that in these combinations, some alleles repress the activity of the other alleles. This is why genetically these 2 are different. This is homozygous, this is heterozygous. When you check the phenotype, they are identical. This will be yellow, and this will also be yellow. And when you check a yellow pea, you cannot say if this is homozygous or heterozygous. I mean, either you have to make a Mendelian cross to understand if it is homozygous or heterozygous, or you have to do DNA sequencing. Otherwise, there is no way of telling it. Okay? Because of the law of dominance, the ratio of genotype segregation is 1:1. So 1 homozygous, 2 heterozygous, 1 homozygous. But the phenotype distribution is 3:1. Okay, this is phenotype, and this is genotype distribution. The 3rd law of Mendel. And at this point, you know what dominance is. At the molecular level, one of the alleles is more active. Either it is transcribed more, or it functions more, or when it has its function even a little bit, it is going to have a greater effect, so it is going to shadow the function of the other allele. So this is the dominance story. So every kind of dominance is different from each other. One dominance can be different from the other dominance, but all in all, in the phenotype, you just cannot say You just cannot see it, like in this case. Green is recessive here. Say, green is recessive. Although these ones, they have this green allele, you just cannot see its effect. Okay. So before we finish, the second law of Mendel, law of independent assortment. And so many of you did it wrong in your lab reports when we were working with Drosophila. So we should go back and check our alleles. Okay. Now you can see A1, A2, B1, B2. Now, if, for example, there is a C allele here, C4 and C5, the segregation of C4 and C5 will be totally independent of A1 and A2. You can have different kind of combinations. You can have the combinations of C4 and A1, C4, A2, C5, A1, C5, A2, because how they align, if they align this way or this way, is a total matter of chance. I mean, they can equally align this way or they can align this way. Okay? So they have 50% chance of segregating together or oppositely. This is called the law of independent assortment. Okay, independent assortment will be very useful for us later. Okay, I'm not gonna go in too much. It's going to take too much time. Okay, independent assortment will be very useful for us. Let's stop here.